Good afternoon. I am Alan Schechter, and I've been asked to make a few remarks about Marshall Nierenberg for this NIH Director's Walls Lecture, one of a series planned to honor Marshall almost exactly 2 years since his passing. Of NIH's five now deceased Nobel Prize winners, Marshall has probably been honored the most of all here. Another manifestation of this is the excellent exhibit on his life and career displayed near the Lipset Auditorium. It summarizes his extraordinary work much more thoroughly than I have time to do now. One amusing point you'll find there is that part of the reason for his coming to NIH 51 or so years ago was that all the lights in Building 10 were on at night, and he took this to mean that all the scientists were so enthusiastic that they worked late in the evening every day. I think the reason for Marshall's lasting impact on the campus are several. Obviously, his deciphering of the genetic code and establishing its universality are the foundations of much of the recent major advances in biomedical research, as Dr. Eric Lander made clear in his Nierenberg lecture last year, and as I suspect today's speaker will also do so. Equally important, Marshall, during his more than 50 years at NIH, showed most clearly that an individual can still think outside of the box and make landmark contributions if his or her institute will support the work. Perhaps Marshall's gee-whiz enthusiasms and his very modest demeanor facilitated this and also led him to be the mentor of an extraordinary group of scientists who made their mark here and throughout the world. It was Marshall, with Chris Anvinson, Julie Axelrod, and a few dozen others, that established the ethos of the Bethesda campus and made it a world-class scientific destination. We are pleased that Marshall's widow, Dr. Myrna Weissman, and many of his former colleagues in the Heart Institute are with us today to continue to honor his memory. Now it's my pleasure to turn the platform over to Dr. Francis Collins, who will introduce Dr. David Botstein, the second Marshall Nierenberg lecturer.
Well, thank you, Alan. Good afternoon to all of you. I, too, am delighted to welcome all of you to the Nierenberg Lecture, honoring this remarkable scientist that we at NIH had the privilege to have in our midst for many years. And I also very much want to welcome Dr. Myrna Weissman, who's here with us, his wife, who has been, all in her own right, a marvelous scientific contributor and certainly a devoted person to Marshall and to his memory, and we're really delighted you could be here. Also glad for former members of the Nierenberg Lab who's able to join us for this. In thinking about who to invite for such an important lecture, it didn't take us too long to settle on today's speaker. And if you read the New York Times yesterday, it turned out to be a feature on the first Nierenberg lecturer, Eric Lander, but it seemed to focus almost as much on the second one, David Botstein, because of the role that he played in recruiting Eric Lander out of, that really boring, abstract area of mathematics into the mathematics that has changed biology, and which David has been such a remarkable leader and advocate for. David is a renowned geneticist, educator. He's now director of the Louis Sigler Institute for Integrative Genomics at Princeton University. But he has traveled through many other distinguished institutions, happy to say one of them including one that I have some affection for, the University of Michigan, where he obtained his PhD, and then with other stints at MIT and at Stanford and at Genentech, ended up in his current role, where he is not only continuing to do fascinating research, but also playing a major role in revitalizing and revamping undergraduate science education at Princeton. I had the pleasure of sharing the Albany Prize with David and with Eric Lander a year or so ago, and that was an occasion to hear some great stories about various things that had happened in their relative careers. I am particularly fond of the story that led to perhaps the paper of David's that had the largest impact on the field of human genetics and set the stage for the Genome Project. And that's a paper in 1980 in the American Journal of Human Genetics, which basically laid out the strategy by which one ought to be able to map any human heritable trait using a panel of variable DNA markers. As I understand it, and this is from David's reporting on it, so you have to take all of this with a little grain of salt. David was attending a training grant review where one of Mark Skolnick's students, I think it was Carrie Kravitz, was talking about hemochromatosis and basically presented evidence that there might be a connection between hemochromatosis and the HLA locus on chromosome 6. And there was a furious debate apparently amongst the immunologists about how hemochromatosis could possibly be an immunological disease, because it didn't seem like that at all. And David and Mark Skolnick, who's in the room, apparently among the few who realized, no, wait a minute, just because it shows linkage to HLA doesn't mean that that is the responsible locus. Maybe they're just nearby. And so David began to try to explain to the assembled group of non-geneticists the concept of linkage, and said something to the effect of, well, you know, HLA is a wonderful locus with lots of alleles, and you know, if we had lots of other loci like that with multiple alleles across the genome, we could map anything. There it was. And apparently they repaired to the bar, which is what one does at moments like that, to try to flesh out a few minor details. And from that came forth the strategy, which made it possible to contemplate taking those thousands of human genes for diseases which had previously been outside our reach, and which many of you in this auditorium have worked on, and come up with an approach that would allow them to be mapped, ultimately identified precisely. And here we are now with more than 4,000 of those human Mendelian disorders having had their loci identified, all beginning with that little conversation at a training grant site visit. So never downplay the significance of the ways in which we all get together as scientists. Some things great will happen, especially if David Botstein is in the room. So we are in for a treat. David is going to talk about evolution and cancer, bringing together two major important biomedical themes into one talk, which I am absolutely sure will be fascinating, maybe a little controversial. We'll have some messages for us all that we'll be thinking about for a few days to come. So please join me in welcoming the Nuremberg lecturer, Dr. David Botstein.
Thank you, Francis. Really nice introduction. It is a funny story, and it really did happen pretty much that way. I wanted to say a couple of words, first of all, about how I got here, the Nuremberg lecture. I didn't know Marshall Nuremberg personally very well. I met him a few times, and I do, however, remember absolutely clearly, like it was yesterday, the first time I heard him talk. I think it was in 1962. I was an undergraduate at Harvard, and he came and gave the talk. Which was the original experiments that led to the identification of, at that time, I don't know, a handful of codons for sure, and a lot more codons. But before the Crick rearrangement of the table, I remember that. It was really a transformative thing for me, because I had just at that point made the decision that I wasn't going to be a physicist and that I was going to do something more like genetics. And here was sort of bittersweet moment, because the thing that actually got me into genetics was the coding problem, okay, which I'd read about in Anfinson's book, and here the coding problem had, you know, a major part of it was solved. I was fortunately sophisticated enough to know that the biochemistry of the names of the codons was just the beginning and not the end of the story, and so I wasn't deterred in any way. But this was one of the great events of my scientific lecture going, I have to say. When I was asked to do this, I was of course honored, and I am honored. And then, as usual, I put it off for a little while into thinking about it, and then finally I decided what I was going to talk about, and I submitted to the people who do this the title, and they didn't want the title. They said, evolution and cancer? We thought you were going to talk about translation. And I thought to myself, what did they mean? Translation as in translational research, or translation as in going from RNA language to amino acid language, okay? Which was the Marshall-Nierenberg part, okay? I actually have no particular interest in talking about either one of those things. I don't work on the latter, and I don't much believe in the former, at least to the degree that some others do. So I decided that I would talk about some basic science. And as I thought about the basic science I was talking about, I discovered actually that it was translational in a way, except that the motivation was pure genetics. So that's what you're going to hear, and I hope you like it. All right. We start with a broad consensus, which I've written up in detail here, about how cancer works. This is the result of the work of literally thousands of scientists in the last 50 years. The most fundamental result of the genome, of course, is the realization that that not only is the code conserved, and DNA is, in all the organisms, but actually that the central functions, at least of eukaryotes, and in fact much further than that, are highly conserved, so that one can reason from one organism to the other much of the time with respect to even very minor details of protein function and what they do for the cell. And the question is, what can we learn about the process of evolution itself from yeast? Because yeast, as I will briefly relate to you, is the source of still the majority of all the annotations of the human proteins. So this is a graph of the functional relationships among yeast genes. Was automatically generated, not by us, but by us in the yeast community, yes, but this is a Toronto operation, Charlie Boone and Brenda Andrews, and this graph was automatically generated based on a single kind of measurement, which is how fast a double mutant colony grows under standard conditions. And then that was put into the computer, and you produce this universe. And you can see the universe has galaxies of different colors. Okay, and the points are genes, and the lines between them are how much one gene affects the other in terms of the growth of the double mutant. Okay? So this is a nice bioinformatics thing. Here's what the annotations are. They're everything you want to know about cell biology. And all the galaxies are enriched for a particular gene ontology annotation. So this is where these annotations tend to come from. When you find something in the vesicle galaxy, the odds are that a gene in there is going to have something to do with vesicle-mediated transport. And if you find one that's in the DNA replication galaxy, this is going to have something to do with DNA replication and repair. Okay? This is all the background. Now, we go into ancient history. Now, some of you will know who Jacques Monod was. How many do you think? Half. Only half. Oh, God. Francis, you got a problem, okay? Only half, okay? How many of you know who it was, Leo Szilard? Oh, almost as many. Wow. That's pretty good. Okay? He was a minor 20th century physicist, for the rest of you, whose main claims to fame was that he conceived of the chain reaction as an atomic and the atomic bomb, and had a patent on both. Okay? Otherwise, he's sort of... When the war was over, he became interested in biology, and he invented this gadget. Monod invented a similar gadget. They were talking to each other. It's not important. It was Szilard. And what this thing is a device for growing cells in steady state. So that what happens is you put fresh medium in, drip, 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 and in the finished culture, which is being mixed, the bacteria or the yeast grow, and they overflow at the same rate that they drip in, just as the diagram would suggest. And in this circumstance, Szilard and Monod were able to show that there's a rate-limiting nutrient, that you can determine what the rate-limiting nutrient is. Could be glucose or it could be anything. And there's also a primary selection. That is to say that as you run this thing, what's going to happen is that if a variant arises in this population in the vessel that can grow better than the others, because growth is exponential, it will eventually take over the vessel. Okay? This thing was devised with this experiment in mind, and we have been exploiting this for the last few years in a try to understand things that were beyond the technology of the ancients, because they didn't have sequencing and they didn't have DNA microarrays. They didn't have a lot of things that they wished they had, but they didn't have. So we did some experiments and published these about a decade ago. And the first 3 experiments that we did looked like this. We grew them for a few hundred generations in this gadget, and then we looked at what was the pattern of gene expression. And what you see here is the standard glycolysis, and what you see is that the green genes are genes that have been downregulated in the evolved strain that we fished out of the chemostat after a few hundred generations, and the red genes have been increased in their gene expression. And this makes perfectly good sense because your textbook of biochemistry says there are 2 ATP that you get from going to alcohol, and 30 plus. It's still controversial exactly how many, under what circumstances. It's changing literally every week, but it's 30-something ATP if you go all the way to the bottom here and nothing is wrong and you have intact mitochondria. Okay, this makes perfect sense. This paper didn't make a very big splash, and it wasn't such a big deal. It was a proof of principle. Here's what some of the data look like in the now standard way of looking at things. And one of the things which you should notice is that all 3 of these independent cultures did about the same thing. They increased all these genes, they decreased all those genes, and they divided the transporters in the same way. Sort of like the Iowa caucuses, right? All right, now, a few years later, my student Maitreya Dunham looked at DNA copy number in these and some more strains that she had made by that time, and she discovered that these things have genomic rearrangements. And in fact, one of the things she noticed was that these genomic rearrangements tend to recur. Does this, for the cancer folks, does this have any resonance? All right, okay. So here we are under selection, and we're making genomic rearrangements. And then if you look in detail, because this is yeast and because molecular biology is kind to yeast, we have a gene resolution on this, even in 2002. And here you can see what's amplified, and here you can see what's not amplified. Here you can see the evidence for translocation. And if you look in detail, you see that there's a transposon right at the point at which the translocation occurs. And if you look in greater detail, you see that the break point must be somewhere right at the transposon. And then if you look at the annotation, here we are again, it's CYT1. And what is CYT1? CYT1 is citrate synthase. It is the first step in the Krebs cycle, which is the gateway between ethanol and the bottom of the cell cycle. And so the translocation has moved some promoter next to a gene that shouldn't be expressed and expressed it. Does that sound familiar? I call this the Palo Alto chromosome because, of course, there's the Philadelphia chromosome. Okay, and the Philadelphia chromosome is exactly the same thing, except we're not talking about, boring old glucose. We're talking about leukemia. It's translational research. Okay, right? Okay? But the process is the same process. And the result is the same result. Okay? It is really the disease of our current way of doing science that we spend way too much time in the weeds and not enough time looking at the forest. Okay? It turns out that if you look at all 8 of those things, and some more, that a lot of the translocations and genomic rearrangements do involve genes that are plausibly involved in being able to grow better on glucose, which was the selection that was applied. Okay. Now, one of the things that you can ask, and this is true, equally true of yeast cultures that are evolving to grow better on glucose or something else, and tumors that are evolving to grow better in whatever niche they began in, the breast or the prostate or wherever, lymph node. And that is, if you do it many times independently, do you always get the same result? Is there a limit of the number of ways in which you can have a cancer or that you can have a better growing population? And one way to look at this is to do the experiment over and over again and do it under different limitations, glucose or phosphate or sulfate. These are all easy to arrange. And then you do patterns of gene expression. And when you look at the patterns of gene expression, you cluster them such that like ends up aligned next to like. And what you see is, well, the red ones, those are all sulfate. The blue ones, there are two radically different ways of doing the glucose thing. And there's a little bit of confusion, but maybe 3 ways. 3 ways to do glucose and maybe 2 ways to do phosphate, maybe 3 ways to do phosphate. There may be some overlap between phosphate and sulfate, but you get the picture. It's not an infinitely large number of ways. It's a few ways. There are a few ways to get from here to there in fitness. Maybe the same thing is true in cancer. Well, actually, we already knew that the same thing is true in cancer, and I will rehearse that for you. First, just to make I rearranged this, and so I'm going to go back to the old arrangement. You can do a nice bioinformatic trick. You can do a Pearson correlation between the patterns and ask the question, how homogeneous are the solutions? And what you see is that for sulfate, you get a single peak with maybe a shoulder, but for glucose, you get 2 peaks and maybe a shoulder, and for glucose, you get 3 peaks, maybe 4 peaks. Okay? So this reinforces quantitatively what I tried to show you qualitatively. The sulfate, it turns out, are always the same. They always start in the same way. They start with an amplification of a single gene, SOL1, which is the major sulfate transporter. This is not a surprise mechanistically, but what's interesting is that in this case, the selection is very strong, obviously, but there are no transposons here to help. This happens by some other mechanism, okay? And we're very interested in that mechanism, but it almost certainly is small amounts of homology of the order of 3-4 bases, okay? You can now do the same thing with tumors. Here's a whole bunch of tumors that have been arrayed in exactly the same way that I did the yeast evolved cultures. And these are all the breast cancers. And I can't help rehearsing the fact that if those of you who still believe in de-differentiation of tumors need to think hard about the question about why all the ovarians and all the breast cancers stay the same. They remember who they are, whatever is happening to them. It's forward evolution, not backward evolution. And if you do the analysis thoroughly, this is of course well known and well published now, it's again more than a decade, you find that there are 3 or 4 or 5, some small number of different patterns of gene expression of breast cancer. So just like evolving in glucose, there's a limited number of ways that you can get from here to there and produce an actual breast cancer that anybody cares about. There may, of course, be 1,000 different kinds of breast cancer that don't cause anybody any problem, and there probably are, But these are the ones that actually are fit enough. And now I should, because somebody challenged me on this during the day, I should make it very clear that when I talk about evolution, I'm talking about the evolution from the point of view of the single cell that started the cancer, and not from the point of view of Homo sapiens, right? Okay, it's cellular evolution. It turns out that if you do this experiment, if somebody else does the same experiment and analyzes the data in the same way, you find that the things that we call the red type and the blue type and the magenta type stay together. And this matters clinically because the survival is radically different depending on which color, which subtype of breast cancer that you have. So again, the analogy is very, very close. And there's another piece to this, which I want to emphasize, which is that we know that there is a class of women who get breast cancer because they have an inherited predisposition. The way to think about that is that the first step in getting cancer was inherited instead of acquired during your lifetime. Okay? And so if that's true, and there are 4 different kinds of breast cancer, then we have an expectation that they maybe don't have 4 ways to produce breast cancer, but only 1 if you start down that particular path. Okay? This is an old genetic idea, and there's a lot of evidence for this in this case. And in particular, you can look at the data I just showed you and simply identify the women who had BRCA1 mutations. And it turns out that they are all the red subtype, what is now called triple-negative breast cancer and what we call basal-like breast cancer. And this is only an anecdote, but it turns out to be true that the BRCA2 predisposes to a different subtype. Okay, you can recapitulate this in mice, and you get the same subtypes in mice. So that makes you think. This is Peru's lab at UNC did this, a very nice paper. And what it makes you think that the way in which the cell is organized in yeast and in the tumor only permits a certain number of pathways to actually get you to someplace useful in the sense that you can outgrow your parents and your competitors. Okay? We're now really in the realm of evolution. Okay. You can continue doing this, and under particular cases, you find another phenomenon which is interesting. And that other phenomenon which is interesting is that sometimes you get both deletions and amplifications of the same segments of DNA. And in this case, this is the general amino acid permease, a very important yeast gene. Actually, Chris Kaiser spent most of his career working on this gene. There are 2 transposon elements, one to the right and one to the left. And what you see is the deletion exactly corresponds to there. If you look, some are amplified, some are deleted. But the really important thing to understand is that there are circles that can and are, as I will show you, made. And these circles are probably on the pathway at least to amplification, and probably also to deletion, because sometimes the deletions revert. Okay? And because we can sequence everything, we can know exactly to the nucleotide or to the blocks of nucleotide where the crossovers occur. And we can actually do standard molecular biology, and this is all published, so you can look it up. Here is the smoking gun evidence that we have circles. These black things could only have arisen if there were circles. And these circles are in vivo. And this data, the fact that these guys don't grow, is very strong evidence that that the gap is active, because dehistidine requires gap in order to kill the cell. Okay. Now, what does this have to do with cancer? Well, The little circles and the amplifications, not tandem, is a feature of very large numbers of tumors. Here's a particular, C-MYC is a particularly susceptible to this kind of thing, and what you find is all these dots represent things that probably arise in a very similar way, where you get a free circle which then integrates or not somewhere else. Okay, now, until now, we have been playing with small numbers. And if you're a serious genomicist, you say, we really ought to scale up, okay? And so Michael Desai and Greg Lang cooked up a scheme for scaling up. And it starts with an observation of Lang's, and pure evolutionary observation, that if you just evolve yeast in a very simple way by serial dilution every day, that what comes up are sterols. Sterile means that they don't mate. And they're sterile because when the alpha pheromone from the other sex comes in, there's a signal transduction pathway that's required in order for the mating to be productive. Now, in order to recognize the pheromone, all of this stuff has to be expressed. And that expression under mitotic conditions is a drag on the system. And what Lang had done was to show that deletions or mutations in any one of these genes that was involved here that shut down the pathway and produce a sterol, they actually have a growth advantage, which you could measure in the chemostat very carefully to be about 2 or 1% depending on the allele of GPA1 that's used. In other words, we have a priori, we know that if we take alpha cells and we do this thing, okay, that what is going to happen, I say A cells, I should say, and do this thing, that what is going to happen is that one of the most prominent ways in which they can grow better is to become sterile. And so the scale-up consisted of exploiting that expectation. And it's based on a theoretical consideration. Now we're really descended into very basic science. We're now going to talk population genetics a little bit. And the Desai and Fisher had understood something which had not really been taken seriously previously, which is that how good a mutation that gives you a known increase in fitness, which we'll call S, how effective that's going to be in the population depends on what the population looked like at the beginning. If the population is very uniform, then what's going to happen is that you're going to get the full benefit of an increase in S. If, on the other hand, there was a big distribution of fitnesses from random mutation in the starting population, then what it mattered where you were when you received this gift of the new mutation. If you were among the living dead in the bulk of the distribution, all that got you is it got you to the nose of the distribution. But if you were in the nose of the distribution, the leading edge, then you were in a position to outrun the whole population and take over the chemostat. Make sense? So what the scale-up consisted of was anticipating that sterols were going to give enough fitness that they would, in fact, become visible in these populations frequently. And what we did was to hook up a fluorescent marker to this thing at the end of this pathway, and if they started out being fluorescent, and when they became sterile, they became dark. And you can separate those populations in a fluorescence-activated cell sorter. They developed an assay, which, as you see, is very sensitive. This is data, okay? So the percent sterile could reliably be estimated from about 10% to about 90%. Okay? And they did hundreds of these. They actually did 600 cultures over 1,000 generations with robotics. Okay? And what you see is that you get many different kinds of trajectories. This is the experiment that we wanted, a real scale-up. So sometimes the sterol appears and sweeps, sometimes early, sometimes late. Sometimes the sterol appears and then is overrun by a competing mutation that is better than it. This was a well-known phenomenon, was called clonal interference. Okay? Sometimes it gets more complicated. There are more mutations, right? And sometimes this was unexpected, but we saw a lot of it, is something which the theorists call frequency-dependent selection, where you seem to get a stable division of the population into two types, the combination of which has taken over the culture and is quite stable. This is just to give you an idea of the scale of success of this experiment. They found very many trajectories. So what can you learn from all these trajectories? Well, one of the things we learned is that Desai and Fisher, which was why they undertook, why this experiment was undertaken in the first place, that Desai and Fisher were right, that if you measure the fitness relative to the starting strain, that's the green line, but in fact, the rate at which the mutants were, the sterols were outgrowing the population was faster than that. And that's what you expect if most of the mutations that we see occurred in the nose of the distribution. Because what you're getting is the red line now is the sum of this sterol contribution and whatever was contributed in the nose. And as you see, that's the general rule. Okay? So this is very nice as theoretical population genetics. And here's another way to plot this. Okay? This is the relative fitness of sterols as a function of the number of generations. These, we assume, were swept by something else, had a very narrow distribution that remains to be shown. The rest of these had, a lot of standing variation to begin with, and the standing variation is really important. Okay? You're all, okay, and this is just a visualization of what happens. If you're in the nose, you win. If you're in the, if you're not in the nose, you lose. Okay? And of course, all of this can be dressed up in a lot fancier mathematics, which I'm sparing. Okay? Now comes the really interesting thing. Of course, we're not done with these, because thanks to super fast, high throughput sequencing and the NIGMS willingness to buy us these machines, okay, for pure science, okay, we are in a position to sequence a lot of these. Actually, we're not in a position to sequence on the scale required, so we've enlisted George Weinstock at WashU, and we're going to sequence, we are in the business of sequencing every 40 generations, all of the trajectories. Okay, and that is a tiny fraction of their capacity. They just sort of seed them into their cancer genome things, and it's part of the microbiome project now. He's supposed to do fungi, so he's got fungi. So now we can actually ask the very obvious question. What is the standing variation? What are all those mutations that put a cell into the nose of the distribution in the first place? Because that's actually the really interesting thing. We sent in the sterol idea as a probe to see what was going on, but what was going on is what's really interesting. So we've started to sequence. We have just a few of these. Now what we're sequencing is not an individual winner. We're sequencing the population. So we're going hundreds and thousands deep at each point. And lo and behold, here's ELO1 and GAS1. Okay? Fatty acid elongation, plausible. Chromatin silencing, plausible for anything. You can explain anything with chromatin. Okay? So this is what you are up against in the future, translational folks, because what we got is sequence, what we got is passengers and drivers, and we don't know which is which. Right? We got a problem. Houston, Washington. Okay? Now, how do you solve such a problem? Well, you understand the theory, and you understand the dynamics, and you understand, what the math tells you. Do you do that in some lady's tumor? No, you can't. Can you do it in yeast? Yes, you can. Is yeast relevant? I leave that up to you. A finer point, we have in this same one, we can actually follow the sterile as well, and we have another, the sterile had a passenger, in this case ERG1. ERG1 is involved in cholesterol, lanosterol biosynthesis, also plausible. But in this case, what you see is that as we go sequencing through, ERG1 doesn't keep up. So the driver is sterile, okay, that's the red, and the passenger is ERG1. And that was expected. If the passenger arrives after the driver, then it can drift. Okay? We have lots of these stable frequency dependent trajectories, and these stable frequency dependent trajectories are really stable. We can perturb them with alpha factor. It's a technical trick of yeast. You don't need to know how that gets done. But what you can see is they generally, but not always, bounce back to give you the same ratio we started with. And it turns out that this is about spatial segregation. It turns out that if you label these, and this is a preliminary result from Desai, who's now at Harvard, and Genya Frankel. Genya Frankel was one of our undergraduates at Princeton, so I'm particularly happy that he's in this. And the guys out here are different from the guys in the middle. Okay? So it's a spatial thing. And the stability is probably, you have to hit them both in order to actually get enough oomph to take over the culture. That happens in tumors too. So the last thing I'm going to tell you is using the chemostat as physiology. And here the idea is you have a rate-limiting nutrient in this, and what we're going to do is we're going to look at growth, and when the thing we try to understand is that at steady state, of course, this part of the equation can be set equal to 0, which means that the growth rate is being set by me. And I can set the growth rate to whatever I want. And it turns out, as Monod had shown in the 40s and early 50s, that the rate at which they grow will be related in the Michaelis-Menten way to the limiting nutrient. If you look at where they are in the cell cycle, you find what Hartwell had originally found, that when they grow slowly, they're mainly in G1 or G0. When they grow fast, they're in other parts of the cell cycle. And that's true regardless. However, if you look at what they leave behind, it's different. In glucose, of course, they eat up all the glucose and they produce virtually no ethanol. In nitrogen, phosphorus, or sulfur starvation, they spare the glucose, and they produce relatively little ethanol. And in leucine or uracil, which are unnatural selections, because these are mutants that can't make their own leucine and are limited on the leucine we give them, which is not a normal situation, they waste the glucose. They ferment it all to ethanol. Does that sound familiar? Well, yeah, it's the Marburg Effect. Right? When you have dysregulation of the cell cycle, you ferment uncontrollably. We can get mutants that don't ferment uncontrollably. They also don't know how to stop the cell cycle. And they die. And when they die, we can get mutants. And when we get the mutants that don't die, we find them repeatedly in the same genes, PPM1 and TOR1. And PPM1 and TOR1 are all involved in this nest of genes that are all related to each other, like in the original Baryshnikov diagram. And these guys are all involved in cancer. These guys are at the heart of the growth control, the metabolic relation of the growth control in cancer. So, the conclusions are there are striking similarities between genetic events and the fitness trajectories that we see in experimental events in cancer, in yeast and cancer. Much of the behavior has to do with the standing variation, which keeps changing. The frequency of occurrence and the stability of mixtures is a feature of both kinds. The number of paths to increase fitness is limited in both situations, presumably by the way the system is wired in the first place. And this regulation of the metabolic control of cell growth results in glucose wasting. In other words, we have many of the most interesting and some of the least understood features of tumors in these simple yeasts. And now a message for the sponsor. Okay? Marshall Nierenberg was a basic scientist, as basic as they come. His discoveries apply to all of biology. And the mechanistic understanding that he provided is the basis. Somebody already said that. Okay. I hope that you've been convinced that our data and their interpretation are sound. And if so, you can see that what we undertook as pure science turns out to be actually really interesting in light of oncogenesis and tumor progression, because we're all eukaryotes here. And as a basic scientist, I've been in this business a while now, pushing 50 years. I occasionally made contributions that found some use in medicine. Never knew in advance which projects would have that effect, and I doubt that Marshall knew, what use this was going to be, except that we're going to understand more. And the point is, basic scientists do what we do in the pursuit of understanding. It's not that we don't care. It's we can't predict. Nobody can. The solution to most problems until more is understood. And understanding is what we do. Okay? And my plea is that the NIH needs to understand this too, and that there has to be a balance between basic science, which gives you understanding, and something that will cure Mrs. Jones's cancer tomorrow. It may not be possible to find something for Mrs. Jones's cancer tomorrow by working on Mrs. Jones's cancer. We might have to be a little more subtle, Subtle is the Lord, but he's not malicious, right? The message for the sponsor is if we starve basic science, there'll be nothing left to translate. That wasn't too tough, huh?
Well, thank you for that very clear message to the sponsor and for presenting an eloquent basic science lecture, but also pointing out its translational significance, which apparently you also believe is kind of interesting.
Yeah.
There you have it, the whole spectrum. And I would certainly like to reassure you, David, that the leadership of NIH, myself included, and all those other 27 institute and center directors, would embrace heartily your arguments about the critical nature of supporting basic science. It is our seed corn. We're not going to know enough with what we've done so far to do very much for a lot of the medical conditions that we earnestly hope someday to be able to manage better. And we don't know from what direction those discoveries are going to come from. So rest assured, there's no crisis here at NIH. At least I would assume from what I know, and I think I know a fair amount, that there is not. So let me ask you and encourage people to go to the microphones to pose questions, because we're webcasting this and that way people can hear the question. I want to press you a little bit about your experiment, which I thought was very interesting, looking at the sterile mutants and finding out what other things are coming along through the sequencing of that, and particularly the way in which you try to tell the differences between drivers and passengers. So, okay, it's easy to see if the passenger arrives later, that you can determine it was the passenger. But as your graph showed, if it arises earlier, then you're in more of a pickle. So how do you propose with this massive data set to begin to make sense of that? Presumably you're also looking for common themes of things that pop up in multiple different cultures, just as we do in multiple different tumors, looking for common themes. But say a little more about that.
Well, we have 600 of them, already, okay? And the good news about 600 is that there will be a lot of data. The bad news is that the data's just beginning to come in, and we expect to be fully underwater. We're standing at the bottom of the Grand Coulee Dam here. We expect to be fully underwater sometime in the middle of the next semester. And then we'll have to see. My approach to this is to say, what we have here is a model system, okay? From the behavior of the model system, we've verified one thing, the passenger and driver before and after, that we had every reason to expect. But in this case, we have, we can look at the kinetics, we can look at the dynamics, we can do lots of things here. I suspect that there are going to be other stigmata of passengerness or driverness that may come up that are in the dynamics of how things work, and we may find other ways to do this. I just want to understand the process so thoroughly, okay, that we don't miss any opportunity to make those distinctions on the fly. I can't do any better than that because I actually don't know how it's going to come out. That's why we do this stuff, right? Yes, sir.
Are your mutations already in the culture when you start the selection, or are they occurring randomly during the selection, or is the selection even increasing your chance of making more mutations?
Yes to the first, yes to the second, doubtful for the third, okay? My friend John Roth has spent the latter half of his career debunking all, without exception, of the attempts to make the third one true. The surprise shouldn't have been a surprise, and I actually sort of kick myself about this. It's not possible to start such a culture, every one of these, without having mutations, without having standing variation. Every one of these cultures started as a clone that was actually made into a single colony by classical methods, by people who know how to do it, okay? And yet every one of them demonstrably has, by the time they grow up to be enough cells to start the selection, to have enough standing variation so that, almost every gene in the genome has been mutated at least once. And if you do the math, that's what you expect. What I kick myself about is I didn't do the math in the first place, and neither did all these other population geneticists. Okay? It's one of those things where after you realize it, you say, oh, why didn't anybody think about this before? And the answer is, we just didn't. Okay, and maybe that's part of the thing. So the answer is the standing variation is there, and it doesn't take very many generations to generate more standing variation, because you get one change per 10 to the 8th every replication. And in the humans, it's even more. I don't read the human genome literature very carefully anymore, but I do know that when you take children and you look at the constitutional DNA, you see fewer mutations than if you take adults, and that shouldn't be a surprise either. And my understanding from the cognoscenti is that those numbers work out pretty much according to expectation also. Mark?
You're making the analogy between a free-living organism like a yeast cell, which is mostly free-living, and a metazoan, which has a body and which all the parts have to work together. So in the free-living cell, we can understand why one cell evolves, just gets fitter, and it outgrows the culture. In the organism, such as ourselves, that result is disastrous.
Yes.
So are you suggesting, A, that this is an old trait?
No.
A sort of vestigial trait of every cell for himself, herself?
No, I think it's no. I think it's an inevitable consequence of the way in which all cells, regardless of whether they're part of a bigger organism or not, grow. So the idea is, it's very Vogelsteinian. The idea is that a cell that is completely under control, has its cell cycle turned off, sits there like a lox on a bagel forever. Okay? Sometimes something happens. Either it's a cell that is normally regenerating, it doesn't see the stop signal by mutation, or it doesn't, it is able to replicate at a very low rate. And at that point, it becomes, from its point of view, a free-living organism in the niche of the crypt.
So
and it's from
similar.
that point of view that your rogue, hyperplastic cell is now a free-living organism. It depends on all sorts of complicated nutrition, VEGF, and God knows what, but those things are available, and whatever daughters of that cell can use that environment better is going to grow more and more, because the original thing has been breached.
Yes, exactly.
Right.
So that's what I said.
Yeah.
So now I would like to ask you to think a step further. What is then the evolutionary significance of cancer? Is it to kill organisms before
I
they
don't
get
know
to
that there is a significance to cancer. I think it's an inevitable consequence of having cells that are capable of growing, and so you've turned them off very many ways. You have very many breaks, and sometimes, over evolutionary time, the number of breaks is not quite enough for people who are old. The evolutionary significance of cancer in general, of why it is tolerated in our evolution, is easy to state. Cancer is a disease of the old, not of the pre-reproductive young, for the most part.
David, you've rightly concentrated on real genetic mutation as the source of the variability that affects the growth here. But
Yes.
we also know from single-cell analysis that there are expression level differences in a so-called clonal population. So if it's a tsunami of data trying to analyze the genomic changes, the question is, how do we get at the impact, since there are roadmap works going on single-cell transcriptomes and so forth, these more transient or temporal changes that affect how a cell behaves at a moment that could give it an advantage that then leads it to a clonal population that then could have secondary advantages going forward.
Okay, I take your point. That's one of the first things that we will look at in the tsunami of data. Because if you're right that there are a lot of epigenetic changes, then we should find things that by quantitation hit in the nose of the distribution, because we can tell, okay, and don't have any mutations that predispose. If there are lots of those, then you win the lottery, okay? If there are none of those, I win the lottery, And if it's in between, it'll be like real life, right? Okay.
So I told you it would be provocative. I wasn't quite aware just who the provocativeness would be directed at, but it has been wonderful having a provocateur in our midst, and let us again thank the remarkable David Botstein.